Today I'll be talking about meiosis in a male fruit fly. I have shown this process through illustrations. Male fruit flies use meiosis to create 4 different sex cells. These sex cells are also known as gametes. We want these sex cells or gametes to be haploids to create an increase of genetic information. Male fruit flies have the diploid number of 8, which makes their haploid number 4. Their haploid number is 4 because the female and male gametes each need to give an even and same amount of chromosomes during meiosis to maintain the same diploid number through generations, in this case, 8. In the end, each gamete will have 4 chromosomes. In meiosis, there are 2 phases, meiosis 1 and meiosis 2. Both these processes are happening in the seminiferous tubes, also known as the testicles, since this fly is male. To begin, I'll be showing this process of meiosis 1, which is homologous pairs of chromosomes separated into 2 cells, which is why meiosis 1 is also known as reduction division. To start meiosis 1 is interphase 1. In this phase, centrioles and DNA are getting ready for division, so they replicate themselves. This process is also known as the S phase. Here I've shown the chromosomes doubling and the centrioles doubling. Now the nucleus and nuclear membrane are beginning to dissolve, indicating the start of prophase 1. The spindle fibers begin to form. The centrioles move to the opposite poles of the cell. As this is happening, chromatids are thickening to become homologous chromosomes and are now becoming visible. Homologous means that they have similar attributes. This homologous chromosomes are intertwining, which creates a tetrad. This is when the process of crossing over occurs. Crossing over is the process of when a paternal, meaning male, and a maternal, meaning female, homologous pairs transfer and trade genetic material of homologous chromosomes. Here I've shown them through stripes. This exchange makes the genes no longer identical and creates more genetic diversity. Crossing over is random and happens in a number of ways. Next to start is metaphase 1. The homologous chromosomes are moved by the spindle fibers to line up on either side of the plate, unlike meiosis, where the chromosomes are on the plate. This is when independent assortment occurs, meaning the pairs of chromosomes are being lined up on the sides of the plate in random order. Again, this is a process used to create even more genetic diversity, and there are so many possibilities. Anaphase 1 is when the spindle fibers pull the homologous chromosomes to the poles of the cell while still staying together. Since the nuclear membrane has broken down, it is beginning to rebuild itself in telophase 1. The cytoplasm begins to furrow, and this is when we see the cleavage furrow appear, and the chromosomes return to chromatid form. In meiosis 1, cytokinesis 1 happens, where the cell membrane splits and the 2 separate cells become 2 non-identical cells. This is when meiosis 2 begins. Meiosis 2 is when chromatids are separated into 2 cells. Meiosis 2 is very similar to mitosis. To begin meiosis 2, interphase 2 happens, where only centrioles are replicated, as we want to keep our DNA even, and an even number of chromatids. This stage is very short. Next, prophase 2 begins. By the nuclear membrane and nucleolus dissolving again, just like meiosis 1, this causes the chromosomes to condense and spindle fibers begin to form, as again, centrioles move to the poles of the cell. This now lets chromosomes have different DNA. During metaphase 2, the chromosomes are moved to either side of the metaphase plate, but this is not independent assortment because only some new genetic information has been added to each sister chromatid. The chromosomes face towards the cellular pole, getting ready to move towards them. Next, we see anaphase 2 occur. The sister chromatids separate and are now put into chromosome form. They are pulled away towards the cellular pole. The same process happens in both cells. Next stage in meiosis 2 is telophase 2. This is when the nuclei rebuild and we see the cytoplasm start its furrowing process again, meaning it is getting ready for its another division. To end meiosis 2, cytokinesis 2 happens, and each cell is separated. We end up with 4 cells having the haploid number N. Again, to restate what I said earlier, the fruit fly is male, so this process is happening in the seminiferous tubules, and the end result will be 4 sperm cells, because the division of the cytoplasm and our male gametes were divided equally. If this was a female fruit fly, this process would be happening in the ovaries. It is called oogenesis. A female will only have 1 functioning egg and 3 polar bodies because during their division of the cytoplasm, it was not equally distributed, meaning only one of the 4 cells got enough nutrients and organelles to survive. The other 3 die off. Thank you.